VIASM several public lectures. As you all know, it is very important for mathematics to try to reach out to the public and to promote the mathematics actually used in many domains of science. They introduced Professor Mesirov from both Institute and Harvard and MIT with fixed machine learning approach for genomic medicine.
So, good afternoon, and I want to thank everyone at VIASM for this invitation and for this nice stay in Vietnam. It's my third visit here, and every visit is more enjoyable than the last visit. So I hope today to talk to you a little bit about the kind of work I do in genomics, give you a flavor of some of the problems and some of the approaches that we use, and what the promise for patient care and for medicine is in the kinds of problems that we deal with and that we solve. So it was just about 10 years ago that the Human Genome Project was finished, that the human genome was sequenced. And that was really only the first step. Because what we got, let me see if I can get it. Where is the pointer on this little thing?
That one.
Is that it? Okay, I got it. All right. So what we got was all the sequence of the human genome, but there was still a lot more to learn. So we want to learn what was in the genome to understand how everyone's genome is different and what those differences give rise to, and to know all the signatures of cellular response, so how the genome is actually functioning within our cells, and even more importantly, what all the significant genomic changes are that are associated with human disease. And in particular, what we're looking for is a more systematic approach to discover and understand the molecular mechanisms of disease, and through that to improve the practice of medicine. So I don't know how many of you remember your molecular biology, so I'm going to give you a 1-minute course in molecular biology and remind you that we're made up of many cells, that in the nucleus of those cells are 23 pairs of chromosomes, and those chromosomes are made up of our DNA. And the DNA comes in 4 kinds of base pairs, A, T, C, and G, and it's arranged as a double helix. That was Watson and Crick's great discovery 50-some years ago. And they are paired. The A's are paired with the T's, the C's are paired with the G's in this helical formation. And in our DNA are the genes. And it's really the genes that are the programs of life. So the genes determine the proteins that execute all the biochemical reactions in our bodies. Now, for each gene, we have 2 alleles. That is, we have 1 copy of the gene on the part of our DNA that we've inherited, our chromosomes that we've inherited from our mothers, and another one on the part that we inherited from our fathers. So that's 2 alleles, and we call that the copy number of the gene. So every gene is supposed to have 2 copies, and this will be important later on. The genes express what's called messenger RNA, and it's the messenger RNA that are the templates for the proteins that do all the work in our bodies. All right, so this is called transcription here, and then translation into proteins. That's all you need to know about biology for this talk, but not all you need to know about biology. Okay, so here's the major question that I'm going to talk about today. And that is how we go from a lot of molecular or genomic data to knowledge. Namely, the same disease in different patients can have quite different results. That is, you can know 2 women who have breast cancer. One will respond to therapy and do very well, another will not, and will have a very short lifespan. So the clinical behavior is different, and the question we ask is, how can we use the molecular signatures of the cell? That is, how can we look at the expression of the genes in our cells and use that to help us understand disease and to better customize the treatment of disease and tailor it to the individual patients. And you sometimes hear this referred to as personalized medicine. So how do we do this? So one of the byproducts of the Human Genome Project was knowing what the genes are, where they are in the genome. And that allowed the development of a technology called microarray technology. And what that technology allowed us to do is probe the cells, and by probing the cells to find out how much of each gene is expressed in those cells. And depending on what cells in your body they are, different genes are expressed at different levels. And depending on whether you're in a normal state or a disease state, also you'll have different levels of gene expression. So with this kind of technology, we can now monitor all the genes in the human genome in a cell at once. So we can get a functional picture of what's going on in our cells. And I won't go through all the details of how we do this, but basically we take a sample, and I deal with patient samples, and we look at we extract the messenger RNA, which remember is the transcribed genome, how the genes have been transcribed, and we use this binding of DNA, where remember the A's and the T's bind together and the C's and the G's, so that kind of what's called hybridization, and you can barely see it up here. I'm sorry this isn't showing up so well. And then we attach a little fluorophore, something that will fluoresce when it's excited by a laser. And then we just stick the RNA from the cell onto this chip. We wash off the RNA that doesn't stick. The things that stick will stick to the genes, and the intensity of the spot on the array that represents the gene will tell us how much that gene is expressed. And actually, if I was talking about details, I'd tell you how there are really 40 of these probes that correspond to each gene, but that's not anything that we need to really worry about. The important thing is that we can really just do one very simple experiment to get a picture of what's going on in all the genes in a cell. And now, actually, because of new technologies in sequencing, and because it's become much cheaper to do sequencing, we can actually sequence the messenger RNA instead of using the chips and get an even better approach. Instead of getting an approximation or an analog picture of the expression of genes, we can get a digital signal here, because we can actually count the number of messages for each gene. So as time goes on, we'll be acquiring the data this way rather than through these hybridization genes. Okay. So I'm going to talk a little bit about 2 different kinds of promise you machine learning. So we're going to do classification, which is a lot of what people do in machine learning. And we're going to look at classification of cancer samples or cancer patients. And they're kind of 2 different types of classification problems. One we call class discovery or unsupervised learning. And the idea here is that we have a bunch of samples. They don't have any particular labels, but we believe that they have some underlying structure. And so what we try to do in unsupervised methods or class discovery methods is to identify previously unknown classes, in this case, types of tumors. In my case, we'll talk about that. And the other kind is what we call class prediction or supervised learning. So here, we have previously recognized classes, and we'll get an unknown sample, and we'll try to place that tumor or that sample into these already characterized classes. And these 2 things work together because often we'll have unrecognized classes, we'll do unsupervised learning to tease them out, and then we'll use class prediction or supervised learning to classify particular tumors. And you'll see what I mean by this right now. So here's one of the first problems that I worked on about, the paper was published 12 or 13 years ago. And the idea here was to identify two different subtypes of leukemia, acute myeloblastic leukemia and acute lymphoblastic leukemia. And here's the two different types of leukemia. So can anybody tell me do you think you could tell the difference between these two? Oh, you could? You just used your mind. Okay, so let me say that this is not a straightforward problem to look under the microscope or look at histology and differentiate between these two diseases. In fact, it took 20 years of basic research to actually realize that there were two different diseases. And the difference is important because you see here that there are two different, very different treatments for the two different diseases. The person who did this work and who discovered the two types of acute myeloid and acute lymphoid leukemia was a man named Sidney Farber, the Dana-Farber Cancer Institute, which is a very, very famous cancer institute in Boston, Massachusetts. It's named after him. So he really did this seminal work here. And as I said, it took 20 years in the lab to figure that out. So this was the problem that we set ourselves, and the idea was if we could solve this problem, it would help us develop a correct molecular taxonomy of cancers, so we'd be able to better distinguish different types of cancers, even within the same general class. And that would let us do more accurate diagnoses, and therefore be able to tailor treatments much better to the specific disease. So here's what the data look like. So, whoops, I keep forgetting that I have this thing. So, each one of these columns, everything in life boils down to a matrix. Applied mathematics, everything is a matrix. So here's the matrix of gene expression. Each of the columns represents a different patient's tumor, and each of the rows represents a gene. And I've only shown you a handful of genes here. Remember, there are something like 20,000 genes in the human genome. Now, where you see a deep red, that means the gene is very highly expressed. There's lots of messenger RNA corresponding to that gene. And if it's low, if it's relatively underexpressed, it's dark blue. All right? And this is the kind of data that we're going to deal with. And the first thing we do is let's look at some ALL and AML patients. So here I've taken the 25 genes that are most upregulated in ALL patients and downregulated in AML patients. So this is called highly differentially expressed. This is how we refer to them in biology. And here you see the 25 genes that are most upregulated in AML and downregulated in ALL. So now the idea is if I have a new patient and I look at these 50 genes, now I can ask, do you think you can diagnose this patient? Can you diagnose this patient? Chow, can you diagnose this patient? Oh, come on. Let's look. These 25 genes look more pink, right?
If I diagnose EBV, then I just die.
All right, anyway, yes. So I can do it. I look at these 25 genes versus these 25 genes, and it looks to me like this must be an ALL patient. And the question is, can I teach a computer to do this? And the answer is yes, I can. So I sort, again, I've sorted the genes by their correlation with the different disease types. And I've also tested their significance. Are they more highly correlated than random? And I use a permutation test to do that. And I use those genes as features in a machine learning algorithm to teach a computer to tell the difference between the two subtypes of leukemia. Okay, so what was the bottom line? The bottom line is that this approach works very well. We use weighted voting, that is, each of those features or genes votes according to its correlation with the disease type for whether the patient is AML, has AML or ALL. But you can use many other machine learning algorithms and basically get 100% accuracy. And In fact, if you took all the labels away and you clustered the data, then you would easily distinguish AML from these two different types of ALL and the two different types of AML from each other. And we've gone on to do so many other types of cancers and also applied it to outcomes, survival, chemosensitivity, diagnosis, and prognosis. Okay, so why aren't we done? Right? It looks like the problem is solved. Well, here is the issue. The reason this was so quote-unquote easy was that there were hundreds of genes that were statistically significantly correlated with one of the subtypes of leukemia or the other. But that's not always the case. So another problem that we worked on was a pediatric brain tumor called medulloblastoma. It's the most common malignant brain tumor in children. And there is a reasonable outcome in terms of treatment. 60 to 80% of the patients are cured. But the cure consists of chemotherapy and radiating the brain. So if you think about radiating a child's brain, a 2-year-old's brain, you can imagine what the side effects are. There's terrible cognitive side effects and developmental side effects. And so the ability to know which children are likely to respond to treatment and not can help you titrate the level of the treatment of these patients. So we tried to use our standard approach, and we sorted the genes according to their correlation to poor and good outcome patients. We had data for those patients, again, using the expression. And the question was, were there any genes that showed a significant difference or were significantly differentially expressed between the two types? And in fact, the answer was no. There were none. So here's a case where the signal is much more subtle, right? So the problem is not totally solved. Nonetheless, we went on and we built a predictor, and the predictor did pretty well. It had almost an 80% classification accuracy, which is actually a little bit better than clinical predictors. That is, if a child came into a doctor's office and he said, this is a good prognosis or a bad prognosis case, based on metastases and histology and things like that. However, the problem was because those genes aren't really significant, they don't give you much insight into therapy, they don't give you much insight into mechanism, and if you take a new data set, it doesn't generalize well. So this is a case of what we call machine learning overtraining. So we could train and do well on a training setting, cross-validation, but not get any generalization.
How do you measure that?
We just see how many errors we make. It's very easy. So in this case, what you do,
How many is or not? Is it something different?
we have clinical outcome data on these patients. So we know that they didn't relapse within 3 years, let's say, or 5 years. So we have their outcome data, and that's how we can measure the accuracy. So that brings up a good point, which is without the clinical data, we can't really do much. So what we learned then is that there's really a hierarchy of problems. That the easiest thing is to tell a diseased tissue from a normal tissue. The next easiest, where you basically get no errors. The next is to tell the difference between subtypes of a particular tumor. And in that case, we have hundreds of markers or significant genes and very low error rates. But then, as we move into things like treatment outcome, then we can have very few significantly differentially expressed genes, and we get much higher error rates. So we know we can build good predictors from time to time for tumor type, for example, to do diagnosis. For treatment outcome, maybe not quite as well. We know there's a hierarchy of problems, but the point is that this classification is really a black box, and what we really want to get is some understanding. So we don't want to just be able to predict the outcome for a patient. We want to understand the mechanism so that we can actually develop better targeted therapies and understand why the tumor is developing or how to stop the tumor from developing. So we need more knowledge. And we're also curious as to why some of these examples don't generalize very well. And the answer is kind of here, that genes are basically not independent variables. And everything I've told you up to this point, we've been using genes as independent variables, but they're not independent variables, they're dependent. Because the way biology works is in pathways and the interaction of genes that are coordinately up and down regulated and so on. So as long as we treat them as independent variables, we're not going to do well. We have to begin to treat them and recognize their dependence. So this is where the notion of using signatures and a knowledge base comes into play. The idea here is that we want to obtain cellular responses and interpret the data that we get in the context of known biological pathways or response signatures, and use that to gain insight into underlying biological processes of disease. So there are a couple of things we have to do that. The first thing is we have to build a knowledge base of these signatures. And so we built a knowledge base of signatures. We call it the molecular signatures database, and it comes from databases of biological pathways or sets of genes, gene lists that we derive from the literature, which have either shown up in certain diseases or known to be associated with certain diseases or known to be up or down regulated after perturbations when a gene is knocked down or a gene is activated, and maybe markers of a particular phenotype. And we have that database completely annotated so that people know what those sets of genes correspond to. And then we're going to use those signatures to interpret array data. And I'm going to talk to you about 3 different ways of doing that: gene set enrichment analysis, a single sample version of that, which we use, and then I'm going to end with this integrated network that we recently published for doing patient stratification, which takes advantage of this signature or pathway approach rather than using the individual gene features. Okay. So what is PSEA? All of that, gene set enrichment and that. Analysis. So the idea is we use annotated gene sets. So sets that correspond, as I said, to a particular phenotype of interest, to a particular biological process that we know of, or a pathway. And just as before, we're going to sort the genes according to their correlation with outcomes, say, the ones that are most highly correlated with poor outcome at the top down to the ones that are correlated with good outcome. Now I'm going to take that set of genes and I'm going to see where does that set of genes lie in this list, which is sorted, remember, according to its correlation with outcome. So if the genes are sort of randomly distributed throughout the list, then we can infer from that 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 particular process or pathway is not really related to the underlying biology of this particular difference that we're looking at. Right? But if those genes are overrepresented at the top of the list, with the genes highly associated with poor outcome, or at the bottom of the list, the ones highly associated with good outcome, then we can infer that that set of genes does have something to do with the thing that we're looking at. And so how do we evaluate that? Well, we use a version of the Komarov-Smirnov test, and we weight it. And the reason we weight it, I'll tell you what the KS test is in a minute. We weight, we use a weighted version because we want to look just not for non-random behavior, but we want to look for sets of genes that are overrepresented at the top and bottom. So that's the reason for the weighting. And then we test for significance using a permutation test. And the reason we use a permutation test is that it preserves the dependence of the genes. So this is the way we do it. Basically, you walk down the list of genes that have been sorted by correlation. And when you hit a gene that's in the gene set, you increase this enrichment score. And when you come to a gene that's not in the gene set, then you decrease it. Right? And you do that walking all the way down the list. You get a little plot that looks like this. And the maximum deviation from 0 is that enrichment score. That's the random walk, and that gives you the Mesirov test. And I've lost the pointer again. Oh, here it is. Okay. And that's the formula. Okay. So the weighting over here is this coefficient rho. If that coefficient were 0, this would just be the standard KS statistic, but it's weighted, and so it's not quite the KS statistic. But again, that helps us distinguish sets that are overrepresented up here or down there. So what happens if we do that in the medulloblastoma data? If we do that in the medulloblastoma data, we look for pathways that are enriched in poor outcome, then what we find, whoops, oh, you can't see it, can you? I'm sorry. On my slide, there are a number of these pathways that are highlighted in turquoise, but they're not highlighted here. So let me see if I can remember which ones they are. So this TOB1 pathway, the MAP kinase pathway, PDGFR pathway, the mTOR pathway, these are pathways that are not only enriched, but they're also pathways which, I guess there's one more, this one down here, RAR, RXR. Those are actually pathways for which that contain drug targets for compounds that have been approved for human use. So the fact that these are targets of compounds that already have been approved for human use means that they can be moved into clinical trials and possibly used as alternative therapies for patients with poor outcome prognosis. So I'm going to go back to this medulloblastoma thing. So again, just to remind you, this first paper that we did, we used expression data alone. We got a classifier that was only about 78% accurate in cross-validation. Does everybody know, I'm sorry, does everyone know what cross-validation is? No, okay. So here's what cross-validation is. In cross-validation, remember, this is what we call a training set. I'm building the model or the predictor using all the data here. In cross-validation, I pull out one poor outcome sample, one good outcome sample. I build a model according to whatever approach I'm using. Then I try and predict those two samples. I do that many, 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 many times. And then I see what my accuracy was based on the clinical outcome data that I have. And that's what gives me this 78%. It says 78% of the time I predict for these two pulled out things. But then remember that you're doing a different model every time because you're pulling out and building a different model based on those individual genes. And that's the thing that didn't generalize. So last year, we finished the study, so that was sort of 9 to 10 years of work, where we sought to do much better than that. And the way we wanted to do much better than that was in a number of ways. Oh, all the colors are going there, okay. By adding more data types. So instead of just using gene expression, we would add clinical data, namely whether or not the tumor had spread to other areas of the body. Histology was someone looking at a slide of the tumor and saying it was one of two types, either classic or desmoplastic. Those are two different types of medulloblastoma tumors. We added molecular subtypes, which I'll describe in a minute. We used pathways rather than individual genes, and copy number. Remember, copy number is the number of copies of a gene that exists in a cell. And what happens in tumors is often instead of having 2 copies of a gene, you you'll have 4 copies of the gene, or 8 copies of the gene, or you'll be missing the gene entirely because you'll have deletions on the chromosomes. So, more data types, identifying and leveraging molecular subtypes for increased sensitivity, employing a hierarchical